Welcome to today's Immune Zoom seminar. It's my absolute pleasure to introduce our speaker, Dr. Manuela Raffatellu from UCSD. Manuela received her MD and MS degree in the University of Sassari, Sardinia, Italy. It's a really beautiful island. And she did a postdoc with Andreas Bauer, first in Texas A&M and then in UC Davis. She started her own lab in UC Irvine in 2008 and was promoted to tenured associate professor. In 2017, Manuela was recruited to UCSD. So from all the places she had been stayed, I could tell Manuela loved warm and the sunshine, so really all beautiful places. So currently, Manuela is a professor in the Division of Host Microbe Systems and Therapeutics and the Department of Pediatrics. Her research focused on the interplay between gut microbiota and mucosal immunity, with the long-term goal of discovering novel and effective therapeutic targets for controlling infections. And she is interested in investigating the host response to gut mucosal pathogens and aim to decipher how the mucosal response to infection is orchestrated, specifically which components are beneficial to the host and which are evaded or exploited by pathogens to cause diseases. And we will hear more details in a second. Because of the extraordinary contributions Manuela made to both the scientific community and the education, she has received numerous awards, which I will not read the super long list. And she was elected to the American Society for Clinical Investigation, American Academy of Microbiology, and the Society for Pediatric Research. And Manuela is undoubtedly an excellent speaker and had been invited to deliver dozens of keynote speeches. So I couldn't wait, but looking forward to your talk today. So Manuela, the floor is yours.
Thank you very much for inviting me and for the very kind introduction. And I'm really happy to be here and to share some of the research that we're doing in the laboratory. So today I'm going to tell you a little bit about one story, but I just want to give first an overview about what my lab does. So we're really working on this interface between pathogens, the gut microbiome and gut immunity, and we look at how pathogens evade host immunity, how host immunity can control pathogens, how the microbiome and pathogens compete. And we look at different cells and the interaction with bacteria and the host in the response to infection. We're mostly gut focused, but today I'm going to also show some lung data actually. So, and the talk that I'm going to tell you today is actually about a chemokine that we've been working with for the past few years. And chemokines don't require much of introduction for this audience, but these are small ligands that they are defined by their cysteines that they contain in conserved position, that they are important for their tertiary structure. And and the receptors for chemokines are these G protein coupled receptor. So the chemokines, so you can see here some example like the CC and CXC chemokines here. And the chemokines and the receptor are involved in numerous processes, from inflammation to homeostasis, to cancer, immune responses to infection and disease and allergies. And immune response to infection is obviously what I've been interested in. So the chemokine that I'm going to tell you about today is a CCL28. Previously known also as MEC, and I started working on CCL28 when my lab was UC Irvine, And because of my interaction with Albert Zlotnik, he's been a chemokine expert all his life through his very distinguished career, both in industry and academia. And Albert recently retired, but I kept, working on this chemokine and continuing the project that we started together. And this work was done mostly by a former postdoc of mine, Araceli Perez Lopez, who's now an associate professor, kind of tenure track still, I think, at the UNAM in Mexico City. And the project was now continued on all the revision, has been done by by Greg Walker, who's a postdoc fellow in my lab. So CCL28 is this mucosa-associated epithelial chemokine, and has a lot of roles, has been fairly well studied, despite the fact that the mouse has not been available until basically Albert made the mouse, and I made the mouse also when I moved here. And so this chemokine is the reason why we became interested is because it's inducible by inflammation and during infection. And when you look at the distribution of this chemokine, so Albert liked to talk about this as a dual chemokine because it's present also in homeostasis and not only during infection. And its expression is particularly high in the large intestine, and is also expressed in the lung, which will be relevant for the second part of my talk. And so Albert also looked at this database and look at the human expression. This is also another way to look at human expression. You can see that the chemokine is very highly expressed in the colon and the small intestine, but it's also present in other organs, very important, the salivary gland, mammary gland, and you can see here also trachea, bronchus, etc. So so basically this chemokine has two primary receptors that have been described in the literature, CCR10 and CCR3. And you can see here that CCR3 is definitely a promiscuous receptor. And this chemokine, so the receptors were studied because basically you can transfect cells with this receptor, and you can see that there is migration when the CCL28 map is added in cells that are transfected with the CCR10 and CCR3. And this, we were interested because this chemokine is induced in the context of colitis. And for example, you can see here that when you look at the colon that is inflamed, these are patients with ulcerative colitis. You can see that CCL28 is highly expressed in the colonic epithelium. And this earlier study here also showed that intestinal the colon carcinoma cells, when they are infected with salmonella, they have an increased expression of this chemokine. So even though there was all this basically work done with the chemokine, there is very little known about the role of this molecule in the context of infection. And I wanted to study this in the context of a pathogen that I've been studying for the past 25 years or more, probably since when I was a medical student. So I'm very interested in non-typhoidal salmonella. These are leading causes of inflammatory diarrhea worldwide. There is over 100 million cases, and in the United States, 1.35 million cases. Drug resistance is an issue, and there is a huge medical cost every year. And also, this is a foodborne infection. So this infection is transmitted through the food source. Classic sources are chicken or eggs. And the disease manifestations are a classic diarrhea. So this is inflammatory diarrhea. However, there is a strong component of host immunity that is protective. In fact, patients with bacteremia, patients with the defects in the immune system, they actually develop bacteremia. And the antibiotic treatment is usually not recommended for patients with just limiting colitis, because by basically depleting the microbiota, prolongs fecal shedding of salmonella. So antibiotic is reserved only for patients that are at risk for more severe disease. And here I just put like a summary slide of our view of how salmonella induces colitis in general and in competition with the microbiota. So we like to think about this immune response as a double-edged sword, because basically when salmonella comes in, it's able to invade epithelial cells, it triggers, it can survive in macrophages, and it triggers the production of a pro-inflammatory cascade, which includes IL-17 and IL-22. And this is important for the recruitment of neutrophils to the site of infection. However, and these neutrophils, we know that they are protective because they can kill salmonella. However, there is also some components of this antimicrobial response that affects the gut microbiota, and salmonella is able to evade these antimicrobial responses and survive. So we've been studying salmonella colitis and its duality for a long time. And so we were interested basically in understanding the role of CCL28 and salmonella colitis. And to do so, we use our colitis model. In this model, basically mice are gavaged with streptomycin, and then they're given salmonella a day after, and throughout the course of infection in susceptible mice, this is up to 4 days. We can harvest the organs and like cecum, colon, Peyer's patches, and then look at also mesenteric lymph nodes, spleen, and liver. And we can do bacterial counts, cellular analysis, histopathology, and we can compare these in wild type and knockout mice. And so I just would like to point out that all of the experiments that I'm going to show you were done with wild type and knockout litter mates. They were co-housed, and these mice, we also looked at their microbiota in general, and this is an example. In their microbiota was fairly comparable, and it didn't really seems to be affected also during infection, which I'm not showing here. So a first experiment that we did together with Albert many years ago basically was simply doing a survival experiment in which we infected wild type CCL28 knockout mice with salmonella and by oral gavage, as I show you. And basically what we find is that at day 3, all wild type mice were alive. So I see something in the chat, maybe. Oh, they're not hearing the voice.
I think your voice is fine.
Okay.
I can hear it fine. Yeah.
Okay, thank you. Basically, and all the knockout mice, they die. And then we also looked at CCL28 in the feces, and you can see that there is, some variation, but there is we're able to detect it by ELISA. And so when we looked at the fecal burden of salmonella during infection, we didn't see much difference at different time points and in between the wild type and the CCL28 knockout mice. However, there was more salmonella in extraintestinal tissue that includes the mesenteric lymph node. There was a trend in the spleen, and in the bone marrow, there was definitely a lot more salmonella in a majority of mice. So these mice were generally more susceptible, which correlated with increased lethality during this infection. And we wanted to see whether this phenotype was dependent basically on a mucosal response or just a generalized response. And so to test for this, we just infected wild type and CCL28 knockout mice with salmonella intraperitoneally, basically bypassing the gut and whatever response was in the gut. And we didn't see any difference in any tissue. Here I'm just showing you 3 examples and indicating that really there was the phenotype that we observed was dependent on something happening in the mucosa. And we looked for different cell types, and considering that in initial studies, they said that the receptor for CCL28 was present on T cells. We looked at the T cells in the gut and comparing naive mice versus mice that are infected on day 2 and day 3. And we did gut, blood, and bone marrow, and it's a bunch of negative data. So I'm just showing you basically the gut and the blood here. Basically, there is no major differences maybe there was a small difference, if anything, increase in the CD4 positive T cells here, but nothing really that stood out in terms of the lymphocytes that are present in the gut or in the blood. And when we looked at the B cells, which are also thought, to be recruited, at least during homeostasis, also via CCL28, we didn't really see differences either in naive or in infected mice. And the receptors for CCL28, at least CCR3, it's thought to be important in eosinophils. So this is actually one of the questions that the reviewers asked. We had to redo all this analysis again because we hadn't looked at eosinophils since in the past, and we can see that basically there is no difference in the numbers of eosinophils that we identified in the gut or in the blood or also in the bone marrow, which are not showing you here. So thinking about the phenotype that we had, increased susceptibility, that made us think that there was a different cell that was involved in the phenotype, and this is actually neutrophils. So what we find is that there is a reduced number of neutrophils that are recruited in CCL28 deficient mice. So when we do this gating strategy, at the end, we looked at Ly6G positive and CD11B positive cells. And there was a significant reduction, and we can quantify this here, and this is the percentage. So these mice have fewer neutrophils, and the neutrophils in the bone marrow or in the blood, they are absolutely normal. So this indicated that it was really something about the neutrophil responses in the mucosa, and there was fewer neutrophils responding to the pathogen. And indeed, once we looked at the pathology, the result was consistent. And in fact, CCL28 knockout mice show reduced cecal inflammation. And you can see here, when we look at the cecum, which is a site with strong inflammatory response scored here by pathologists, you can see that there is a lot more cellular infiltrate in the wild-type mice than in the knockout mice. And something that we noted is that there is in particular two pro-inflammatory cytokines that seem to be reduced, at least at this time point, in the CCL28 knockout mice that are IL-1 beta and interferon gamma. And this has been actually consistent across two postdocs, and a few years apart, they found exactly the same result. We think that this is actually important for the difference in pathology that we observe. So CCL28 knockout mice have reduced neutrophils. As I mentioned earlier, neutrophils are important during salmonella infection and are protective. And we know that they are recruited very early in response to infection, in particular via the IL-17 pathway and the production of 6 chemokines, 6CL1 and 6CL2. So that results in neutrophil recruitment. So we wonder basically what if CCL28 was modulated at the neutrophil recruitment, retention, activity, and basically we looked if there was expression of the receptors of CCL28 in neutrophils. And when you look at blood or bone marrow neutrophils rest basically in uninfected mice, most neutrophils actually don't express the receptors, which is probably why for the longest time people didn't think that neutrophils would respond to this chemokine, because at rest this receptor is not present. However, the situation is very different once we isolate the neutrophils from the gut of mice that are infected with salmonella. And so basically here again we use our streptomycin model, then we do necropsy and we take the intestine, blood or bone marrow, and in all cases, especially in the mucosa, in the gut, we see now an upregulation and induction of these two receptors, CCR3 and CCR10. And so that tells us that there is something about infection that is inducing the receptor on the surface of neutrophils to respond to this chemokine. And indeed, once we take bone marrow neutrophils in this case, and we stimulate them with pro-inflammatory molecules, now we see surface expression of these receptors. In particular, CCR3 has been the strongest receptor. CCR10 is a little more, there is a less induction. And you can see here that we can either use a cocktail or pro-inflammatory cytokine, interferon gamma, TNF alpha, GM-CSF, or FMLP, or PMA, that is a very strong stimulant, or also bacterial LPS. To different level, all of them basically promote the induction of CCR3 on the surface of neutrophils. And we know that phagocytosis also enhances the expression, because if we take bone marrow neutrophils and we stimulate them with pro-inflammatory cytokines, and then we add the beads, then you see here that there is an enhancement. Actually, I think actually here I copied the wrong graph. It looks the same. Sorry. This is particular. This is true definitely for CCR3. I might have picked the wrong one here for CCR10. But you can see that there is an induction basically of this receptor. And during salmonella infection, actually, so in this case, we did a slightly different experiment. So we got the bone marrow cells, we enriched for the neutrophils, and then in vitro, basically, we infected these neutrophils with the salmonella at an MOI multiplicity of infection of 10 bacterium per cell. And then we looked at CCR3 expression. And you can see here from uninfected up to 2 hours, and even 4 hours here post-infection. But basically, you can see that it's quite shocking, because by the time we had salmonella, it just takes 1 second. And in like 5 minutes, the time to go in and fix the samples, there is already receptor being expressed. We kind of start seeing strong expression at 1 hour, really post-infection, and then by 2 hours, it seems that it reached the peak. So definitely it's consistent with the fact that phagocytosis seems to be an important signal for induction of this receptor in neutrophils. And indeed, we can see that this is dependent on phagocytosis also during salmonella infection. So here we picked a 1 hour time point, and can see basically that there is CCR3 expression in neutrophils, and then we can block this by adding cytokalasin D, which basically blocks phagocytosis. So again, it's consistent with our assays with the beads showing that phagocytosis is important for inducing expression of this receptor. We also wonder whether CCL28 plays a role in neutrophil chemotaxis. And to do this experiment, we use basically transwell with the chemokine that is put in the bottom chamber and activating neutrophils in the top chamber. And then for 2 hours, we evaluate migration. You can see here, this is the no chemokine control, that neutrophils migrate very well in response to the known neutrophil chemoattractant CXCL1. And they do exhibit some migration in response to CCL28. However, it doesn't seem that its migratory function is as strong as the known CXCL1, the role of CXCL1. Also, there are some studies that think that chemokines have a direct antimicrobial, they propose a chemokine having a direct antimicrobial activity, and some certainly do, especially at the high concentrations. However, salmonella is fairly resistant to antimicrobial peptides, and so and is also fairly resistant to CCL28. So here we use bacteria like 10 to the 5 times 10 to the 5 bacteria with either no chemokine or increasing concentration of chemokines. of chemokine, and we did basically direct bacterial killing, and you can see that Typhimurium salmonella wild type is fairly resistant, and so but the concentrations that we measure in the host are about here actually, so fairly low, but even at the highest concentration is fairly resistant. And we compare wild type salmonella to a salmonella mutant that has an altered outer membrane and is more susceptible. And also E. coli K12, you can see here there is a big difference with salmonella wild type. So even at the highest activity, CCL28 does not appear to have, at the highest concentration, a strong antimicrobial activity. Direct. So, however, what we find is that the CCL28 enhances neutrophil antimicrobial activity against the salmonella. So in this assay, we we basically culture, we put these bone marrow neutrophils and we infected with salmonella, and then with or without CCL28 as the chemokine. You can see here in the non-chemokine control, or a chemokine by itself, again, at the particular concentration does not have an effect on salmonella. There is no neutrophils here. So when neutrophils are added, there is, this is the salmonella survival at 1 hour in neutrophils. There is some killing at least, you know, and some by some neutrophils. However, killing is definitely enhanced where CCL28 is added. And this is largely dependent on CCR3 because when we add a CCR3 antagonist, this is a molecule that has been developed to antagonize basically this receptor. Then we basically rescue salmonella from the CCL28 dependent neutrophil antimicrobial activity. So here we're showing that this appears to be important. This appears to be important in vitro. So how do neutrophils kill salmonella, and what is the role of CCL28 in enhancing killing? And one of the things that we find is that CCL28 increases neutrophil ROS production during salmonella infection. So neutrophils make reactive oxygen species to kill bacteria and fungi. And you can see here that infected neutrophils, they make a little ROS. They make more when they are infected with salmonella, and even more when they are when there is a CCL28. And what we find is that, again, this is primarily dependent on the receptor CCR3. Here, for this experiment, we used, so here you can see the neutrophils were stimulated with CCL28, and these are uninfected. These are infected with salmonella and either treated with an isotype control or an anti-CCR3 antibody, and you can see that we have reduction of ROS. The effect of CCR10 was more variable. We only see it actually really for one, and but there is also potentially some effect from CCR10 on this, but definitely the CCL28 effect was bigger. The other mechanism by which CCL28 helps neutrophil activity is also via the formation of neutrophil extracellular trap. Initially, this data has been done by Araceli, my former postdoc Araceli, in her own lab with human neutrophils that were activated with platelets. And so here you can see that they unstimulate neutrophils. They don't really make nets. So this is with the DNA stain. And you can see that when their platelets are added, neutrophil make nets. But when on top of platelets, CCL28 is added, there is a lot more nets formed. And this again is dependent mainly on CCR3, because when she uses the CCR3 inhibitor, now there is a reduction in net formation. So what I show you so far is that the CCL28 plays a protective role during salmonella infection. So our view is basically that when salmonella comes in and triggers this pro-inflammatory response, neutrophils are recruited via CXCL1 and CXCL2 and the CXCR2 receptor to the site of infection. And in the gut, there is a CCL28 that is enhanced during infection, and neutrophils in response to salmonella, phagocytosis, and also the pro-inflammatory environment enhance expression, particularly of CCR3, but also in part CCR10, and this result in a protective effect, in protection against enhanced protection against salmonella infection. We were very excited about this because we find a lot of host responses that are evaded by salmonella, and it's very rare that we find something that is actually able to provide protection against this pathogen. So we were very excited about this result. We were so excited that we wanted to see whether this is applicable also in the context of other infection. And so basically I spoke with my colleague and division chief, Victor Nizet, here at UCSD, and we wanted to look at the different pathogen in the different body sites where neutrophils were also important for the infection control. And so we chose Acinetobacter baumannii. This is an environmental bacterium that is really a huge problem in healthcare settings, because even though there is not very many cases, it's highly lethal. And it can cause a wide range of infection, including pneumonia, sepsis, urinary tract infection, etc. And this strain and this bacterium is very highly resistant to antibiotics, and in fact is considered as an urgent threat by the CDC. And if you can see here, there is also a huge medical issue in medical cost and that. So we decided to collaborate, as I said, with Victor Nizet and perform a lung infection of mice with Acinetobacter baumannii, and 3 postdocs in his lab in different times helped with this experiment. And I was very concerned when we started this experiment, because in this infection, basically mice are injected in intratracheally with Acinetobacter, and all the wild-type mice, this is a lethal infection, they die, basically most of the mice die within 2 days. And considering that the CCL28 knockout mice died so quickly during salmonella infection, I was afraid that we were not going to get an experiment here, considering how quick is the phenotype and the lethality phenotype in these mice. However, I still remember the day in which Araceli basically did this infection, and what she found is that basically CCL28 knockout mice are protected during lung infection with Acinetobacter. She came back and told me that they were all jumping up and down, happy. And at the beginning, we didn't know what to think, but we have an explanation, and I'm going to show you. And this was actually a strong phenotype, and we're very happy because considering how problematic this infection is, the fact that we have a potential way to modulate this infection is also very exciting. And we didn't really see differences in bacterial burden in these mice anywhere. So not in the bronchoalveolar lavage, in the lung, or in the blood. The colonization seems to be similar. So we decided to look at the cellular infiltrate in these mice. What I can tell you is that the proportion of cells in the bronchoalveolar lavage, fluid, and lung, blood, and bone marrow, so the neutrophil proportion was similar, but the number was different. And in fact, the live cells, and you can see that basically most of the cells in the bulb, as Victor promised me when we chose this model, are neutrophils. And in the CCL28 knockout mice, we had fewer neutrophils, which is basically the same thing that we saw during salmonella infection. And then we did all the cell typing also here. And my postdoc, I really like how we put this data, because I think it makes it very clear how neutrophils basically comprise the vast majority of cells in the bronchoalveolar lavage, and there is other cells, but they are a real minority. And there is a nice difference, probably about 2 to 4-fold difference between wild-type mice that are infected with Acinetobacter and the CCL28 knockout mice that are protected. They have fewer neutrophils, and this is protected in the context of this lung infection. And this is also visible at the histopathology, because when we do staining for anti-Lys6G for neutrophils in the lung, so you can see here, this is a beautiful uninfected lung from both the wild type and the CCL28 knockout mouse. When they are infected with Acinetobacter, you can imagine how this mouse has difficulty breathing. There is a lot of cellular infiltrate here. And in contrast, the knockout mice have fewer neutrophils, and their pathology looks better. And so how are the neutrophils from the Acinetobacter infected mice? These neutrophils also express CCR3 and to lesser extent CCR10. And also, so again, indicating that there is, during infection, there is an expression of the higher expression of this receptor. And also in vitro, this is a slower kinetics, but for about 4 hours of infection with Acinetobacter, the neutrophils are actually also in vitro expressing CCR3 like they do during salmonella infection, although with salmonella it happens within 5 minutes. With Acinetobacter, we have to wait a little bit longer. And also, in contrast to salmonella, CCL28 does not enhance neutrophil antimicrobial function against Acinetobacter. So whenever we incubate Acinetobacter with neutrophils, here is without the chemokine, and this is with the CCL28, there is really no difference in antimicrobial activity against Acinetobacter. And so this, and in fact, this is consistent with the fact that we don't see differences in the CFUs anywhere, and we think that CCL28 is providing protection here because of the reduced number and potentially activation of neutrophils that causes less tissue damage, which helps the mice to eventually survive. In summary, we think that we are uncovering a role of CCL28 in response to mucosal pathogens, and this seems to be an important chemokine that contributes to neutrophil trafficking to the mucosal sites of infection, and this seems to be primarily due to CCR3. And these neutrophils that we isolate, particularly from the gut and the lung mucosa in these 2 infection models that we used, they express the CCL28 receptor, CCR3 and partly CCR10. And CCL28 seems to be particularly good at enhancing neutrophil antimicrobial activity against salmonella. Acinetobacter seems to be resistant, and we don't know why and whether this is, for example, strain dependent. I'll show you the infection is very severe when Acinetobacter makes it to the lung. Basically, we see different outcomes that are depending on the pathogen infection and the different body sites. And we think that this is due to the fact that we're balancing the neutrophil antimicrobial activity and tissue damage, that if there is control of infection, at least we can tolerate some tissue damage. But in the case of Acinetobacter, there is just this massive tissue damage by the recruitment of neutrophils that end up being detrimental. And I can tell you that Araceli is doing some work related also with other lung pathogens, even with the flu, and seems to be, similar. And also something that I don't have too much time to discuss, but I'll be happy to take questions. That CCL28 was also one of the chemokines that were found to be induced during COVID. And again, we think that that probably has something to do with the tissue damage in response to the infection that was due to the excess neutrophil. So this is kind of our working model that, you know, you have neutrophils in the bone marrow and they don't really express much of these receptors on their surface or in the blood. But then when there is an infected lung or gut mucosa, then there is the CL28, and in response to the pathogen phagocytosis and the pro-inflammatory cytokine environment, there is an increased expression of the CCR3 and to lesser extent CCR10. And this leads to neutrophil activation, and the final outcome is host protection or host damage, depending basically on the pathogen, the tissue, and so we think that this is a key mechanism that we could potentially use for modulating neutrophil function. So with this, I would like to acknowledge my laboratory here. As I said, Greg and Araceli really did outstanding work with this project. As Araceli is now at DUNAM, and she also continues to contribute from there. Other people in my lab also contributed, master students, Steven Silva, Karina Melchior, and Romana Gerner. Collaborators Albert Slotnik and Victor Nizet, and these are some of the funding sources for this project, and I'm really thankful to the support from the NIH, Burroughs Wellcome Fund, and Kenneth Reinin Foundation. And I'd like to thank you, and I'll be happy to take any questions. And actually, I'll leave the model because I think it's probably more productive for a discussion. So thank you very much.